slides again?
Yeah.
Yeah. Okay.
Just verbalizing that people are being recorded, because I was told once that you're supposed to say it after you hit record too.
I'm
So
glad you thought
we're
of that,
recording.
Fox, because I would not have. All right, I think we're set to go. Good morning, everybody. I noticed the other day on the news, and I bet a lot of you did too, that there was a terrible, harmful algal bloom off the southern coast of California, I think it is now. And it is wreaking a lot of damage to wildlife out there on the coast. And so today's talk, I think, is especially timely. These red tides are increasing in intensity and frequency on the West Florida shelf in the Gulf of Mexico as well. These blooms are thought to be fueled by nitrogen originating from coastal sources, but identifying the specific sources has been pretty challenging. So it's a real pleasure to introduce our speaker today, who has looked at what drives these harmful algal blooms. Rene Boiteau and his colleagues have developed an approach to fingerprinting organic matter, finding that groundwater is a significant source of organic nitrogen during a major bloom event. And when I had talked to Rene about this, and he was talking about groundwater as a source of nitrogen, I was just immediately interested, and I thought we should really hear about this. This research provided new insights into the role of organic matter in transporting key elements like nitrogen, and perhaps to better understanding the drivers of algal blooms. Rene completed a PhD in chemical oceanography in 2016 from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution, and did postdoc research in the Environmental Molecular Science Lab at the Pacific Northwest National Laboratory. After research at the College of Earth, Ocean, and Atmospheric Sciences at Oregon State University, he moved to the Department of Chemistry at the University of Minnesota in 2023. Rene, it is great to have you with us. Take it away.
Hi, Mark. Thank you so much for that introduction, and thanks for the invitation. So as Mark mentioned, I recently moved, well, a year and a half ago to the University of Minnesota from Oregon State University. And so part of my motivation of giving this talk is also to make connections with you folks at the Minnesota Pollution Control Agency to see how our research might be able to contribute to understanding of pollutants and contaminants that impact the state. So the focus of this talk is going to be on our project on the West Florida shelf, trying to understand the sources of nutrients that fuel ecosystem production in the Gulf of Mexico. And this has important links to red tide events and harmful algal blooms. Before we get into that, I'd like to introduce my lab and give a bit of background on the types of approaches that we bring to this problem. So the general goal of my lab is to understand how organic matter in the environment influences the fate of nutrients and metals. And we do this using approaches that we develop combining liquid chromatography that can separate different molecules that comprise organic matter in the environment, which is a complex mixture that incorporates carbon, but also other nutrients and even metals. And then we use mass spectrometry to detect the individual molecules that are present in that mixture. And that allows us to track thousands of different chemical species at the same time and identify what nutrients and elements are associated with them. And this, as Mark said, it has been really useful for fingerprinting different types of organic matter that have different effects. And so in general, we're interested in taking these tools and applying them to problems like understanding nutrient availability, which is the topic of this talk. Where are nutrients coming from and how do they affect how does the form of those nutrients affect the organisms that can use those nutrients? In other cases, we're interested in contaminant mobilization, so I won't talk about it in this talk, but organic matter also affects how much heavy metals can be removed from wastewater and things like that, how much metal is migrating through the environment and how it's taken up by plants. And so another thrust of our research is focused on understanding that process. And then also, a lot of these organic molecules can be useful geochemical tracers of different physical or biological processes. Right. And so, in general, our goal is to develop this molecular picture, right? So I like to compare it to, looking at through a microscope at a microbial community in the ocean for the first time, right? And so what you start to see is that there are lots of different species, looking down the microscope, that are doing different things. Some of them might be primary producers that are photosynthesizing, others are breaking down carbon. They're interacting, and with these speciation tools, we can identify the major forms of elements that are present in the environment. We can start to categorize them into key groups, right? And then we can start to develop overarching rules that can describe how they interact and how they contribute to the way that the system functions. Okay, so one of the first studies that we did looking at organic matter was really trying to understand what drives the persistence of organic matter in the ocean. And so this figure here is showing a schematic of the state of knowledge of different reservoirs of carbon with a focus of organic carbon on the planet. This is really important because organic carbon is a major buffer of atmospheric CO2, right? If you organisms, plants take carbon dioxide, fix it into biomass, right? That removes it from the atmosphere system. It's the fate of that organic carbon that dictates over long time periods how much carbon is in the atmosphere. So we can see here that there's a lot of carbon in the atmosphere, and that's growing 870 petagrams. If we look in the ocean, huge amounts of carbon are present in the ocean as organic carbon. And this figure here is breaking that up into different classes of carbon. We have biota, the organisms themselves, but that's actually a very small amount of the carbon that's in the ocean, only about 3 petagrams. About 10 times more carbon is present as particulate organic carbon. So this is mostly biota that has recently died. And this is important because it's a larger fraction of carbon, and then this is also material that can sink through the water column and transfer that carbon from the surface to the deep ocean. And then even more carbon, the vast majority of the organic carbon in the ocean is present as dissolved organic carbon, right? And the amount of organic carbon in the ocean is roughly similar to the amount of carbon in the atmosphere. And so it's really important to understand what are the drivers of this concentration and how could they change over time. So this figure I also like because it shows that we normally think about organic carbon as boxes, right, of homogeneous material, but of course that's not true, right? So we can see that organic carbon here has a lifetime that can be estimated by radiocarbon measurements ranging from about 0 to 10,000 years. So some of this carbon might be very, very recent, other carbon might be very, very old, right? And different forms of organic carbon are likely to be cycling on very different rates. So people have thought about this for a long time and have tried to infer, what are the different components of organic matter in the ocean that turn over on different time scales. And so when people have started to map out the concentration of organic carbon in the ocean over time, and over time and space, we kind of developed this picture that there are essentially 3 different reservoirs of carbon that turn over on different frequencies. So this profile here is showing dissolved organic carbon concentrations that were measured on a cruise that we went out in the North Atlantic, just off the coast of Bermuda. So this is really in the middle of the North Atlantic Gyre. Well, not the middle, but within the North Atlantic Gyre. And you can see that there are higher concentrations of organic carbon in the surface compared to depth. And colleagues like Craig Carlson at University of California, Santa Barbara have measured these over and over again at the same spot. And they find that in the surface, we have these elevated concentrations. You can see I've colored this section in blue. And that material, if you look across the season, disappears and regrows in seasonally. And so we know that the time scale of that carbon is turning over on the order of months, right? We see that if we go deeper in the water column, below 200 meters, which is about the depth that sunlight penetrates, we can see another elevation of carbon. And we can see that this carbon gets removed over the timescale of ocean circulation. So this carbon, this fraction of carbon is elevated in places where this deeper water forms. And if you track that water as it migrates through the deep ocean, you can see that it's removed over the timescales of hundreds of years. right? And then everywhere you measure dissolved organic carbon in the ocean, you find concentrations that are at least 40 micromolar. And if you go into the very deep ocean, where the concentration is about 40 micromolar, and take the radiocarbon age of it, you find that its average age is around 4,000 years. And so we have this idea that this is organic carbon that is refractory. It can't be used by organisms, and it's just cycling around the ocean multiple times, and it's well mixed everywhere in the ocean. Right. So again, this gives us this idea that there's different, we can measure dissolved organic carbon, but it really doesn't represent one thing. It represents components that have different lifetimes, right? So, and understanding the chemical origin of that is really important, but it's hard because these are very complex mixtures. And so this is a problem that we have started to address using our liquid chromatography mass spectrometry approaches. And so, we actually go out on ships and we collect samples. So this is what this looks like. This is actually from a cruise off the coast of Oregon, but we have these rosette samplers that have bottles that are basically tubes with a top and bottom that can be cocked open. And then this has an instrument package on the bottom. And so when we deploy the instrument, we bring it down to the bottom of the ocean. We collect information about the salinity, the temperature. We know the depth that we're at, chlorophyll, which we use as a proxy for algal abundances. And then when we bring the package back up, we can trigger the bottles to close at different depths to capture that water. And then we can bring it back on board to sample and characterize. Okay, so shipboard collection, we go out on ships, we collect samples with these deployable samplers. And then when we're characterizing the organic matter, we have to concentrate that and remove salts. So we do a solid phase extraction on board, and this basically pumps the water through. We filter it so that we're only looking at dissolved carbon, which is the majority of the organic carbon here. And then we capture that organic matter on solid phase extraction columns. And so this captures the majority, not all of the organic matter that's coming that was present in that bottle, and concentrates it on that column, and we flush the salts away. And then back in the lab, we can take these columns, elute them with methanol, and then we have this mixture of the organic matter that's been isolated from different depths or different locations in the ocean. Okay, so then we can take that solid phase extracted seawater and we analyze it by liquid chromatography with ultra high resolution mass spectrometry. And so those complex mixtures are then being separated. There are tens of thousands of different molecules that are abundant in any given sample that get separated, and then we can detect the individual molecules by electrospray ionization, ultra high resolution mass spectrometry. So the ultra high resolution of the instrument gives us the ability to differentiate molecules that have very, very similar masses. Some of these molecules deviate by extremely small mass differences, like 0.001 daltons. So we need these very powerful mass spectrometers to be able to distinguish them. The other advantage of these instruments is that the mass accuracy of these spectra are extremely accurate. And so what that allows us to do is assign confident molecular formula to each of the molecules that we've detected. And so we've worked really hard on developing algorithms that allow us to take this data and basically for every molecule that we're detecting, we can assign a molecular formula to that. And that's helpful because now we can say, how much carbon that molecule has. Does it have other elements like nitrogen or metals associated with it? And then because we're distinguishing these different molecules and able to quantify their relative abundance across environmental gradients, we can use this to start to fingerprint organic matter that has different sources or different cycling patterns, right? And so we can start to group molecules that have similar spatial distributions, right, to reduce the dimensionality of this data, and then that gives us these ideas of components that are either present from different sources or have different removal properties. Okay, so applying this to this open ocean system where we have this idea that in the surface we have the semi-labile organic matter extending down into the mesopelagic about 800 to 1,000 meters, we have this semi-refractory dissolved organic matter, and then mixed throughout the water column, we have this refractory organic matter. We can see that the molecules we detected fit the same exact pattern. So this plot here is showing the relative concentration, the relative abundance of individual molecules that we've detected and annotated with the molecular formula across the samples we collected, which are ranked from depth. So we have the shallowest samples at the top and the deepest samples at the bottom, ranging from 5 to 1,000 meters. And the very bottom is our blank. And then the heat map here is showing warm colors, these lighter colors indicating higher abundances and the cooler colors indicating lower abundances. Right? And so using clustering algorithms, these 3 different clusters emerge. And one of them is present primarily in these surface samples down to 200 meters. This matches exactly where we expect to find this posited semi-labile dissolved organic matter. Another cluster is present in the surface at higher abundances, but extends into these deeper waters. This matches the distribution that we expect from this inferred semi-refractory dissolved organic matter. And then we have one component that was present throughout the water column, refractory dissolved organic matter. And so this was really, really exciting to see because these ideas that you have organic matter that is cycling quickly in the surface, less quickly into the mesopelagic, and then very slowly, well mixed everywhere, is really just inferred from measurements. And here we have really some of the first molecular evidence that these fractions really do exist. And we have an idea of the molecules that are present in each of these different components. And that allows us to distinguish their chemistries, right? So we know the molecular formulas of these different molecules, and we can compare them broadly across these groups. And we find that their semi-labile dissolved organic matter tends to have higher hydrogen to carbon ratios. It's more reduced, less oxidized molecules. They're also larger molecules. We know the mass to charge ratio. And then the retention time that we're doing chromatography is telling us something about how hydrophobic they are. So more hydrophobic compounds are eluting later. And so we see that this more labile carbon has a more hydrophobic character, and then the refractory organic matter is more polar. And so this starts to give us a picture of what are the chemical properties that lead to persistence, right? And we came up with the schematic showing that you generate this very complex distribution of thousands, tens of thousands of different organic molecules in the surface. And then faster, less oxidized molecules that are larger and more hydrophobic are more rapidly removed, and we have the persistence of these smaller, that more oxidized molecules that likely have higher water solubilities. And so we kind of have this idea that persistence is probably largely driven by the tendency of these molecules to form aggregates, right? Things that are removed faster are probably less water soluble. They'll aggregate, and that can either create a hotspot for microbial degradation, or if the aggregate gets big enough, that could actually sink throughout the water column. So again, this is showing us how understanding the chemical properties that distinguish these different components can help provide insight into the processes that are controlling organic matter concentrations in the ocean. Okay, so we had the idea that perhaps we can actually use this approach to fingerprint nutrient sources in the ocean. So I focused more on bulk carbon in the first study in the North Atlantic Gyre, but we ended up proposing getting funded to do another project that was trying to look at what are the sources of nitrogen to the coastal ocean. And our focus was on the Gulf of Mexico, with the rationale that this is a place where these inputs are really important because they can fuel things like harmful algal blooms. And the inputs really are not well constrained, right? So if we look across the ocean, most of it is nitrogen limited. This plot here is showing sea surface nitrate concentrations. Warm colors are indicating high concentrations, and cool colors are indicating low concentrations. And so we can see the vast majority, about 2/3 of the surface ocean, has extremely low nitrate concentrations. And that's because organisms are basically taking up that nitrate until they run out. And then that's what caps productivity. So if we look in the Gulf of Mexico, right, these purple colors are indicating, this is a place where nitrate is extremely low. And so further production and things like algal blooms are really directly driven by the inputs of additional nitrogen. Okay, so this the idea of blooms in the Gulf of Mexico is well known in that community because of the effects of red tide. So this is just an example of a handful of articles that have highlighted the harm of these blooms. Right, in 2018, New York Times had an article on a giant 10-month-long harmful algal bloom that killed huge amounts of marine life in the Southwest Florida coast. A couple of years later, there was another fish kill from another summer red tide event. Just 2 years ago, when we started our project, there was a huge red tide event, and this was also highlighted by the local media. Right, the communities are heavily impacted by these events because they are health hazards to humans, and they kill a lot of wildlife. So this is a picture of the beach in St. Petersburg, Florida, after one of these red tide events, and you can see all these dead fish that have been poisoned by the blooms. So harmful algal blooms are really a big coastal issue all across the Florida shelf. This picture here is showing the West Florida shelf. So we have the Gulf here, showing the circulation currents. So water comes into the Gulf of Mexico through this loop current. So it's basically North Atlantic water that comes in. It has extremely low nitrogen concentrations. And then it loops around. So it comes up and then passes going southward across the West Florida shelf. And you can see in red here, all of the spots where red tide has been observed in the eastern Gulf of Mexico between 1950s to 2014. So pretty much all along the coast, but really this hotspot region along the southern shelf. Right. So as I mentioned, the issues associated with these blooms are fish and marine mammal kills. They also they're also associated with respiratory irritation to humans. And then if humans also eat shellfish or swim in those waters, they can ingest poisons that can have really bad health effects. Right. So there's been a huge effort in Florida to basically better predict and understand what's driving these events, with the goal of trying to provide better information to people about when they might happen, and ultimately, ideally, we can mitigate the problem itself. Right. So red tide itself is driven by Karenia brevis. This is the bad actor in this scenario. So Karenia brevis is a dinoflagellate that can produce this potent neurotoxin brevetoxin. So this is one of the structures of brevetoxin. This is really the reason why these algal blooms have a lot of health effects. This is a microscope image of Karenia brevis. When it blooms, you can see these giant red sheets in the water if the density of the cells are high enough. And so, the problem with Karenia brevis is and red tides is that not only are they an issue, but they're becoming more and more frequent and intense. So this figure here is from a paper that looked at the severity of blooms between 1953 and 2018. And you can see in these stacked plots, bloom severity index is on the y-axis versus year on the x-axis. The severity is broken out by counts within certain binned classes. And so we can see in these later years, there's far higher, there's a big increase in the number of blooms and their intensity, right? So these dark red bars are more and more intense over time. So why is this becoming more frequent, more intense? We don't actually know, right? And so this is a complex and multifaceted problem, and I'm sure those of you in the audience can probably appreciate that more than even I can as a mostly analytical chemist. But we've worked closely with a range of biologists and physical oceanographers and chemists to understand the context of this. And so the goal of our study is really to understand one of the drivers of these blooms, which is ultimately the source of the nitrogen. Right? So Karenia brevis cells are basically present everywhere along the West Florida shelf at low concentrations. And then at certain times of year, they will bloom, right? And so this can be driven by some physical processes, right? Providing a seed population, maybe temperature and light is another factor. But the bloom cannot happen unless there's nutrients that are fueling it, right? So the source of nitrogen is really important. And then another thing is that the source of nitrogen has to end up being taken up by Karenia brevis. So the Karenia brevis is also competing against other algae for these inputs. And this is particularly true of diatoms. And so in some cases, you get blooms that are diatoms, which are not harmful. In other cases, you get Karenia brevis. And the reason for this isn't always clear. One of the curious things about Karenia brevis is that unlike diatoms, it can actually assimilate a lot of organic forms of nitrogen, in addition to the nitrate and phosphate that's broadly available to most algae. Okay, so the source and the specific types of nitrogen that are coming into the West Florida shelf to fuel these blooms is unclear. But there are a bunch of hypotheses that have been proposed to try to explain them. Okay, so hypothesis one is that red tides are getting their nitrogen from upwelling. So this figure here is showing the concentrations of nitrate and nitrite, it's mostly nitrate in the open ocean, from samples collected in the Gulf of Mexico, as well as other places in the Atlantic Ocean. The Gulf of Mexico, I believe, are these orange points. And so there's this consistent trend where in the surface you have very, very low concentrations because that nitrogen is being taken up. And then as that organisms that have accumulated that nitrogen die, they sink, and then that nitrogen, organic nitrogen, is remineralized into nitrate. And at some point, that nitrate can come back to the surface to fuel more production, right? The concentration really doesn't start rising until you're several hundred meters below the surface of the ocean. So one of the original theories was that there were observations that red tides are often really intense after hurricanes. And so scientists suggested that maybe the hurricanes are stirring up the water, driving upwelling, bringing nutrients to the surface, along with Karenia brevis cells that are living in those deeper layers. And then that upwelling can drive the blooms. When people have looked at this in more detail, it seems that most of the upwelling water is only occurring from about 150 meters and above, and the nitrate concentrations here are just too low to account for the nitrogen demand of the blooms that are observed, right? So this might be a partial contributor, but it's probably fairly minor. Okay, hypothesis two is that the source of nitrogen is actually in situ nitrogen fixation. So certain organisms can take nitrogen from the atmosphere and turn it into bioavailable forms. And in the ocean, and in the Gulf of Mexico, the most important organism for this is Trichodesmium. So this is a filamentous cyanobacteria that can also fix nitrogen. Trichodesmium fixes about 50% of the nitrogen across the global ocean. So it's a super, super important organism. And there have been some observations that Trichodesmium blooms are in the Atlantic Ocean and in the Gulf of Mexico are linked to Sahara dust events. So Trichodesmium needs to fix carbon, photosynthesize, and it needs to fix nitrogen. Both of these processes require a huge amount of iron as a catalyst for the enzymes that are conducting these reactions. And iron is very insoluble in seawater. And so if there is a source of iron, that can fuel the growth of organisms like Trichodesmium. So dust coming off of the Saharan desert deposited in the North Atlantic or the Gulf of Mexico is often followed by a bloom of these nitrogen-fixing bacteria, and then those nitrogen-fixing bacteria are providing new nitrogen that could potentially fuel blooms. And so in some cases, there have been correlations between the severity of dust deposition in the Gulf of Mexico and the intensity of red tide blooms, leading to this hypothesis that maybe there's this ecological succession. Dust drives nitrogen fixating trichodesmium, and then at some point the Karenia brevis population uses enough of that nitrogen and can bloom. So the estimates of this process could explain potentially a large amount of the fixed nitrogen that's needed to explain the blooms. The problem with this hypothesis, though, is that it's unclear that that nitrogen would actually be taken up by Karenia brevis rather than have other fates. And it doesn't really explain the timing and the spatial distribution of those blooms. So in the plots that I showed earlier, most of these blooms are really concentrated across the coast, whereas the Saharan dust is being deposited throughout the Gulf of Mexico, and most of the trichodesmium are really offshore as well. So again, this is a potential hypothesis, but seems to have some holes that can't explain most of the distributions and frequencies and intensities of red tides that are actually observed. So hypothesis 3 is that the source of nitrogen is rivers. So if you look at the concentration of nitrate and organic nitrogen in rivers, it's much higher than in seawater, right? And we can see that water is flowing into the ocean. And so this certainly is a source of nitrogen coming into the ocean, but if you quantify that flux, again, there just isn't enough nitrogen to explain the blooms, right? It can account for about 17% of the nitrogen needed for the algal blooms in many of the studied years. And it also can't generally explain the spatial distribution of red tides. So this figure here is showing some of the river watersheds in pink here and then in green. So a lot of the water is flowing into Tampa Bay, right, and coming out, and then that water is being flushed south from the loop current. But a lot of the red tides are concentrated either much further south of this or further north, right, where that river water nutrient isn't getting, right? So again, this is probably part of the source of nitrogen, but generally, doesn't account for everything or even the majority. Okay, so the last hypothesis, right, is as Mark alluded to, is actually the one that we think is probably unrecognized in importance, is groundwater. And so, river water is fairly easy to study because you can see the river, right? You can easily sample it. You can measure the nitrogen phosphorus. You can measure the flow rate very easily, and you can determine how much that water is coming into the ocean. Groundwater is another avenue of entry for terrestrial waters to end up in the ocean. So, water flows on land, and then some of that ends up funneling into rivers, but others percolate through the ground, and then that water can come out into the coastal ocean through submarine springs, right? And that groundwater, when it's entering the ocean, ends up being a mixture of some of this fresh water that's discharged, that's taking terrestrial nutrients and organic matter, things like that, and bringing it into the subsurface, as well as some marine water that's being entering the ground and then coming up in a mixture of that fresh water through these submarine springs. Okay, so the interesting thing about submarine groundwater is that it's anoxic. There's a lot of organic matter in these sediments, and so oxygen is used up. And then we normally think of these anoxic groundwaters as places where nitrogen is being removed. So, when groundwater comes into an anoxic condition, then if there's still a lot of organic matter, it will be broken down through by using nitrate as the terminal electron acceptor, and eventually that will be taken all the way to nitrogen gas, right? So we tend to think about these places as places where nitrogen is actually being removed from the system. But it turns out that in these systems, because there's no oxygen around, these other pathways for breaking down organic matter are a lot less energetically favorable. So you have much higher concentrations of organic matter, and a lot of that organic matter has nitrogen. So most of the nitrogen that's present in these groundwaters is actually not present as nitrate, but as some ammonia, and then the majority as organic nitrogen, right? Because there's just not enough energy for microbes to break off the nitrogen from those molecules. When that nitrogen ends up in the ocean, then when there's oxygen around, that can be further processed, and then the nitrogen can be released and used as a nutrient, right? So the problem with studying groundwater is that its sources are diffuse, you need more intentional studies to focus on it. So one of the goals of our study in 2023, that started in 2023, was to differentiate between these potential different sources with really a focus on trying to understand the submarine groundwater discharge end member, right? Is this actually a potential source of nitrogen that's coming across the entire West Florida shelf, right? And if so, can we track it, and can we end up getting at fluxes and see if they can explain the demand for these algal blooms? Okay, so we had a series of cruises. The main one occurred in February of 2023. And this was a time where there were very intense algal blooms along the West Florida shelf. So this picture here is from the Florida Fish and Wildlife Conservation Commission. They monitor algal blooms, and so they have very frequent sampling. They get cell counts, and then they make this information publicly available, right? And so here we see these red points showing really, really like extremely high concentrations of Karenia brevis. These are the places indicating spots where you have those classic red tide, red waters, and really big health problems associated with them. Okay, so during this time, we conducted a series of field campaigns to constrain the sources of nitrogen to the shelf. Where is the nitrogen fueling these blooms actually coming from? Okay, so this is showing our field site. So if you see here, we have Tampa Bay sort of in the middle of the shelf here, where we have a lot of these algal bloom, these high cell counts. Here is a picture of our field site. So Tampa Bay is here, and then all of these blue spots are places where we took a ship and collected discrete water samples that range from the surface all the way to the seafloor. And then these triangles are places where we took river water samples. So we sampled a series of rivers across the shelf. And then in collaboration with the US Geological Survey, we were able to collect water from submarine groundwater wells. And so the USGS has mapped out a lot of the places where water is coming through, coming up into the ocean from underground springs. So Chris Smith at the USGS installed wells and sampled them in these 3 locations. So we could actually get these groundwater M members, right? So altogether, we ended up with about 175 samples. This was a really big project that involved a bunch of collaborators. We were just one of the groups. Our work was focused on understanding the molecular composition of the dissolved organic nitrogen from these different end members and then tracking them across the field. We had other colleagues, Angie Knapp at Florida State University at the time, who was measuring total dissolved organic nitrogen concentrations. Drew Chappell was measuring trichodesmium abundances. Joe Tamborski at Old Dominion University was measuring radium isotopes, which are useful tracers of groundwater. And we'll talk about that more in a bit. This here is just a picture of these underground well sites. Okay. So, we have hundreds of, close to 200 samples. We conduct our liquid chromatography and mass spectrometry analysis. This is showing a similar heat map to what I was showing before, but you can see that it's a lot more complicated when you're in this coastal environment. You can't just break this out into 3 groups. There's multiple groups. So we use k-means clustering to generate groups of dissolved organic nitrogen that have distinct distributions, and then we can take the most abundant of those groups and start to try to figure out where they're coming from. And so the most abundant group was a group of molecules that was basically present at the same concentrations everywhere, right? This is that refractory organic nitrogen. And that refractory organic nitrogen background in the ocean is a really big reason why it's hard to track organic nutrients coming into the ocean, right? You have to distinguish these organic molecules that are coming in that are actually then being utilized from this massive background of organic matter that is essentially not used up by the microbial community. Right, so small differences in the overall concentration of organic nitrogen can still represent large amounts of nitrogen that are affecting the ability of organisms to grow. So of these fractions that we were able to identify from clustering that are associated with coastal inputs, this was the most abundant one. And I'm showing here a section plot of its relative abundance across this region here. So moving from left to right, we're going inshore and then across the shelf. And so we can see that the abundance is really high as you get to those shelf stations. And then if we compare that distribution to the distribution of radium-224, we see that there's very closely matching distribution. So radium is produced in the ground from uranium decay. And so as groundwater picks up nutrients and things like that, it is also picking up radium isotopes that are being generated constantly by uranium is just slowly decaying in the rocks of the sediment. And radium-224 has a very short half-life. So when you measure its concentration in the water column, you know that that radium has come recently from the ground, right? So the half-life is only 3.6 days. So after a couple of weeks, all this radium is basically gone. So we can basically use this as an indicator of where groundwater is coming from, and so this gives us a pretty strong indication that nitrogen coming in from the coastal coming into the coastal ocean has this radium source. And then to further corroborate this, we had the groundwater end members, and we're able to see that the composition of the nitrogen in that end member is similar to the composite we're seeing the same molecules in that end member as we are in this fraction of organic nitrogen that's being detected across the ocean. This is showing the abundance of that fraction of nitrogen across the surface, just looking from the top. And so we can see here, again, these circles are indicating places where we also had submarine groundwater wells. And so those are hot spots of that this fraction of organic nitrogen, again, consistent with the idea that this nitrogen is really coming from those groundwater sources. And then we can see that throughout the shelf, that we can track and detect that organic nitrogen, right? So this is again further evidence that groundwater is supplying a major component of organic nitrogen that we're seeing across the shelf. Because we have these radium isotopes, we can also estimate not only how much groundwater has been mixed into those coastal stations, but how long ago it left the ground. And from that, we can actually estimate the time scale of removal of this organic nitrogen. And so by comparing the abundance of the dissolved organic nitrogen to the removal rate of the radium isotopes, comparing basically long-lived radium isotopes that give us a conservative tracer of groundwater and short-lived isotopes that give us a time scale of removal, we can estimate that the organic nitrogen coming from the ground is being removed on the order of 22 or 23 days, right? So this again is really exciting because it means that not only is the groundwater a likely source of dissolved organic nitrogen, but that organic nitrogen is being removed, right? And likely that's basically being taken up as a nutrient. It's not just refractory organic nitrogen that's going to persist in the water column. Okay. So the connection to harmful algal blooms at this point is still something that we can only infer, right? So, but we can see here that Karenia brevis cell counts are also distributed at the same sampling time across the shelf in the same sort of region where we see the groundwater derived dissolved organic nitrogen. So these cell counts were collected by the Fish and Wildlife Research Institute. They deployed a glider, which is basically like a robot that can zip around and collect cell counts. So they were not necessarily getting cell counts at the same place at the same time as we were collecting samples. So the spatial, there's some spatial differences in the data here. But in general, you can see that cell counts were elevated all throughout this band, where we also observe groundwater. So at least there's the potential, right, for that groundwater derived organic nitrogen to be a source. Right, so we're still conducting analyses and generating data from this cruise. Our next goal is to basically take dissolved organic nitrogen concentrations and use this molecular data and tracer data to help define different end members and come up with a budget for how much organic nitrogen is coming from the different sources, rivers, the groundwater, and then I didn't talk about it here, but we also have ideas of nitrogen source from trichodesmium, and then the goal is basically to see how those fluxes compare, right, and which ones seem to be higher magnitude, which would suggest that they're more important sources. So again, I started my faculty career at Oregon State University in the Department of Ocean Sciences. It's been really wonderful to move to the University of Minnesota. And one of the things I've really loved about Minnesota is, starting to think a lot more about terrestrial systems and lake systems. And so I'm so thrilled to be here talking to you guys at the Minnesota Pollution Control Agency, because I'm very interested in seeing if we can take some of the approaches that we develop in my lab and start applying them to some of the problems that Minnesota faces, right? So, doing a lot of searching on your websites and looking at papers, I know that, harmful algal blooms are a big problem here, right? Especially microcystis, right? So this is a map of microcystin toxin concentrations detected across Minnesota, right? This is a publication from some of you at the Minnesota Pollution Control Agency, right? The upshot is, half of the lakes that were sampled had high concentrations, right? And so this is a pretty big problem. I wonder, right, what are some of the challenges in predicting the harmful algal blooms that are occurring here? How well are the sources of the nitrogen and phosphorus that are fueling these blooms known? Is groundwater an important source, right? Or is organic nitrogen coming in at certain times a potential driver of some of these blooms? And so, I would love for any of you that are interested in see some connections here to reach out to me, and I would love to have some follow-up conversations. This is, again, something I'm still learning about, but I would love to work on this more going forward. So just some quick summary points. Submarine groundwater distribution appears to be, a potentially important source of organic nitrogen to the West Florida Shelf, and this could be one of the drivers of harmful algal blooms that had been previously unrecognized, right? Because it's mostly this organic nitrogen coming in that's hard to differentiate from refractory organic nitrogen in the ocean, right? So, in the next period of our project, we're trying to put all the data that we have together to quantify the fluxes of nitrogen coming from these different sources and see which ones are the most significant contributors. And so I'd really love to thank my research group who worked on this. Alana Farrell analyzed all of these samples, and she, along with my former lab manager, Kate Kuba, and Peter Bright, collected these samples. Saini Huang and Nicole Coffey have been supporting students in my lab that have also helped with this project. And then all my collaborators on this project, especially Angie Knapp, who was our fearless chief scientist leading the cruise efforts, Joe Tamborski, who did the radium measurements along with his student Andrew, and then Christopher Smith and Salvo and Hannah collected a lot of the groundwater samples that enabled this. And of course, I'd like to thank funding. This is funded through the National Science Foundation and Science Foundation. And with that, I'd love to take some questions.
Thanks so much, Rene. I'm not currently seeing anything in the chat. if people have questions, as usual, feel free to write them in the chat or, raise your hand, unmute.
While we're waiting for people to put the questions in the chat, I am sure we're going to have them. That is an absolutely amazing piece of work, Rene. That is really interesting. I do have a comment question.
Yeah.
In the work that I've been doing over the last 25 years or so, I've been focused primarily on the role of nitrogen in groundwater as an electron acceptor, right? Nitrate, and then other things like sulfate and CO2 and so forth, and iron. But what I'm hearing from you is that, we have in that anaerobic environment, the fate of nitrate, for example, like you mentioned, goes away, but we might be well ignoring the role of this inorganic nitrogen or this organic nitrogen that is moving to groundwater, which is a whole nother redox environment, and then has a different fate. Then it's being used in a different way than the electron acceptor of nitrate in groundwater, right?
Yeah, absolutely. So, the idea that groundwater it is an important source of nitrogen is somewhat counterintuitive for that reason that you described, right? Like nitrate is removed from these anaerobic zones. But if you look at organic carbon, right, organic matter is much, much higher in concentration in these groundwater system, in anoxic waters. And as you mentioned, a lot of that organic matter contains nitrogen, right? Yeah. So then, once that organic nitrogen is introduced into an oxic environment, right, it can then be broken down, and then, the microbes are different. I mean, we're still learning about, clearly the nitrogen that's accumulating in these anoxic systems is very different than the nitrogen that accumulates in oxic systems. And once it enters oxic systems, we see that it's removed. So still trying to understand exactly the fates of that carbon, but it's likely, fueling some productivity.
All right, we have a question from Jennifer. Do you think you'd be able to apply your carbon fraction approach to quantify the different pools to inland waters?
Yeah, we are actually trying to do that. So we have another project at a USGS site in North Dakota, the Cottonwood Lake site, and there we're trying to look at the processes that stabilize carbon in prairie pothole wetlands. So there are a bunch of different wetlands at this site that have slightly different chemistries, and they also have different amounts of carbon storage and organic carbon. And so we're starting to do some seasonal surveys to basically look at, how does the organic carbon in pore waters and in the surface waters of these wetland ponds differ over the course of the year, with the goal of trying to see, okay, do we see that there's some fraction that's turning over relatively quickly? Do we see some fraction that's liberated during, at certain times? And then, that will give us a sense of, what are the different fractions that are cycling at different rates? And ultimately, that project aims to use that information to inform models that are trying to, break out organic matter in the system into different components with different lifetimes. So yeah, that's definitely something that we're interested in.
Oh, another one. Robert asks, are you engaged in any work concerning source characterization of deposited materials, examples, organic carbon and sediment cores, or strictly from water samples via your LCMS approach?
Yeah, thanks again for that question. So as I mentioned, we are at this Cottonwood Lake site. This is another big collaboration with Department of Energy National Labs. And so there we're bringing a bunch of complementary techniques to bear on this problem. And so the LCMS approach is really useful for characterizing this water soluble dissolvable fraction of carbon, right? And there's some relation between that and carbon that's being absorbed or aggregated and stored in sediments. And so for that project, there are some complementary techniques that are looking at the solid phase, mostly synchrotron-based approaches, and then also some pyrolysis gas chromatography approaches. So we can see if some of the patterns we see of different fractions of organic matter in the dissolve phase across these different lakes mirror fractions of distinct organic matter in the same systems in the solid phase. It's definitely challenging though, connecting the solid and dissolve phases. Yeah.
Follow-up question from Ada. My understanding was that the organic matter tended to adsorb to soil. Where is the organic matter coming from?
Oh, this is a really great question too. So, yeah, so a lot of the a lot of organic matter does adsorb to soils, and like mineral phases in the soils that can be liberated at certain times. So right now we don't actually necessarily have a great idea of specifically where the organic matter is coming from. In those groundwater systems, you have a lot of organic matter that is being produced in the overlying waters that's sinking to the sediments, right? And so the sediment surface has a lot of organic matter, and that's breaking down, and some of that is becoming dissolved and then can be flushed back up. Then some of the fresh water that's coming in is also contains organic matter, dissolved organic matter, right? And perhaps that's, contains organic nitrogen that's coming from agricultural systems or things like that. So, this project didn't really set us up to figure out really what the origin of that organic nitrogen is. It's a more zero order question of like, is there organic nitrogen coming out from these systems? But then understanding, can we track the sources? Is it from the sediment surface, or is it from further inland? Is possible because we know the chemical characteristics of the nitrogen and we can track it inland. But thanks for that question.
Emma asked if SGD is found to be a significant nitrogen source. Has this correlation between groundwater and HABs raised any ideas of how to mitigate red tide from this source?
Yeah, again, a really great question. So it kind of relates to the previous question where, like ultimately, where is that nitrogen coming from? And it could be coming from further inland, which could be driven by agriculture, right? I mean, this is where a lot of the nitrogen that's coming into the rivers is coming from. And so, if you can understand, where, if it's inland nitrogen, if it's coming from specific anthropogenic sources, right, then we can perhaps think, identify those. And then, of course, there's a lot of that needs to be done to reduce nitrogen utilization, which is complicated, but that would be possible. It's also possible, though, that the nitrogen is actually just coming from naturally occurring cycles, and so the frequency of increased frequency of these events might just be driven by more intense processes that are supplying more of this groundwater to the surface. But if the nitrogen is just cycling it naturally, right, that could be actually the driver of the red tide. So along the coast of California and Oregon, for example, red tides are driven by nitrogen, but probably most of that nitrogen sources, but most of that nitrogen source is probably not anthropogenic. And of course, with these natural cycles, then it's harder to figure out how to mitigate them. And what we can do is improve our predictions of them. And of course, as temperatures continue to warm and climates continue to change and rain patterns continue to change, right? We want to be able to predict how those effects are going to result in more or less intense blooms or change where the blooms might occur.
All right. Thank you so much. It is 10:02, so I think we are probably going to cut it off there. I want to be respectful of everybody's time this morning. Thank you so much, Rene, for the incredibly interesting talk. It sounded like a lot of folks were, very interested in this. Rene did put his email in the chat. If people are interested in reaching out, that is there. And once again, thank you so much for your time this morning. We really appreciate it.
Thanks again. And again, I really would welcome some follow-up conversations. And yeah, who knows, maybe this can turn into some ideas for LCCMR funding or something else where we can actually work together. So thanks
Yeah,
so much
absolutely.
for the attendance.
Thanks so much. Have a great rest of your day, everybody.
Thanks, everyone.
Thanks again, Rene, for giving that talk. It was really, really interesting.
Oh, thanks a lot.
And
Yeah, it's a lot of
we had a lot of good interest too. A lot of people tuned in.
Yeah, it's definitely a project where we're still working on it and the results so far have been super interesting and exciting, and I think there's a lot of really nice work to build off of. But it'll be cool to see the